Hello, everyone.
Hello.
Good morning. Whoops. I may have pushed myself out. All right. We're going to do things just a little bit different today regarding signing in to chat. If you would, type your first name in, and if you have any kind of a nickname, go ahead and type that in for me too. If there's a nickname that you like to go by, I'd be happy to know that. All right, let me get this started here. All right, very good. That's great, thank you. All right, we're just at 11:20, so I'm going to go ahead and get started. Today, we're going to be talking about the topic that we began last time together, which is the topic of aseptic technique. And we're also going to begin a discussion of a very popular differential staining technique called gram staining. So we won't finish the gram staining material today, but we'll get started on it. We can finish it on Friday. But before we get into the lab material, I just want to let you know that as of this morning, I have received all of the lecture exams. So everyone has taken the lecture exam, which is great, including all the makeup exams. So everybody's got their exam in, which means I can get those graded and get them back to you. You can expect to have your grade back on Friday, probably early afternoon on Friday. So keep an eye out for that. Anything else? Does anybody have any questions if you've started into the prokaryote lecture topic for the week? Does anybody have any questions so far about that material or any other material that we've been talking about? We can certainly talk about that today as well too. Remember, as you're watching lectures and taking notes for yourself, if you have questions about what you're watching or reading or anything, just jot them down for yourself and bring them into our Zoom meetings and we can certainly talk about them. And remember too, you can also post questions on the discussion board that's set up for questions. And you can reply to each other on that board as well if you're trying to figure out something from the lecture material. All right, very good. So the last time we were together, we were talking about this group of standardized sort of methods that we use in the laboratory that we refer to as aseptic technique. We talked about some of the general things that we do in order to be using our very best aseptic technique. And then we started into a demonstration of some of the very specific things we do in the microbiology lab, things like how we would move cells from one broth tube into a fresh tube of broth, how we would flame the neck of a test tube, a glass test tube, of course. And then we finished up last time looking at how we would take cells from a broth tube and use them to inoculate, that's the term we use, inoculate a Petri dish that had agar in it. So that's where we'll be picking up this morning. Give me just a moment and I'll share my screen with you. So right now you should be looking at an image that shows the incinerator on the left and a plate, an auger plate here in the middle, and my hands, I'm holding an inoculating loop. This is right about where we left off. I'm going to overlap a little bit that you've already heard, and then we'll finish up. We have about 10 minutes left in this video. I'm going to take my loop. Remember, incinerate, sterilize, get rid of those cells. So essentially what I've done is I took a bunch of cells out of the culture tube, the broth, and I placed them onto the first place that I hit on that auger. I put a large number of cells, a large amount of cells onto the surface. And then by sliding my loop across the surface, back and forth and back and forth, what I'm doing is I'm spreading those cells across the surface of the plate. So that I'm giving them room to grow on that nice nutrient-rich solid media. Let's take a look at that again. Plate comes up, loop touches the media, and then without lifting it, I'm going to just streak it out, streaking it back and forth in a zigzag motion, spreading out those cells. As we go through the summer and we look at other techniques that we use in the microbiology laboratory, we'll also look at different ways that we can streak an agar plate, depending on what we want to achieve in terms of the growth of the cells. Now, anytime you open a container that has growth media in it, nutrient media or any other kind of media, remember what we said. You want to keep that container open for as short amount of time as possible. You want to quickly get it inoculated with cells, and then you want to close it again because you want to prevent contamination. Now, different people use different techniques to achieve this. You can open and close an agar plate a couple of different ways using aseptic. Here's an agar plate that's being held right side up. And in this technique, we're going to use the lid to keep the plate partially covered. You see that? I can reach in between with my loop and do my streaking pattern, close it, and then put it upside down. Let's look at that again. I'm going to keep the plate right side up, use the lid as partial protection from microbes falling onto it, streak it, close it, back upside down. Now let's take a look at a couple of ways that you don't want to handle your Auger dish. First of all, you never want to just open up an agar plate and hold it straight up to the air, like this. You're just asking for contamination to fall onto that plate. So don't do that. You also don't want to do the other extreme, which is keeping it completely upside down and trying to inoculate it. You will push your loop into that auger if you do that. What you want to do is open up the plate and hold it at an angle like this. You can get access to the surface. You can get your streaking done. Get the plate closed. You're good to go. The last thing I want to talk about today isn't directly related to aseptic technique, but it's the tools that we have available in addition to what we've talked about already. These are single-use, disposable inoculating loops. They come in a sterile package of about 50. When you open up these packages and you pull out the loop, you can see the handle end is what's going to come out first. So you widen up the mouth of the bag and you carefully pull this out, and you've got a sterile loop at the end. Now, some people don't like these single-use inoculating loops. They are not nearly as flexible as a wire loop. And obviously, they're a single-use plastic item. So you can imagine that when they're being used in a busy microbiology lab, hundreds of them are being used every day and discarded into the landfill. So some people are opposed to their use. It's a problem in all of laboratory work that we use a lot of plastic. We use a lot of single-use plastic, disposable materials. And hopefully, at some point, we will get away from that. But this is a tool that's available to us. And some people really like these because it speeds up their work. This loop allows you to do a single transfer of material and then just toss it into the autoclave bag. You don't need to take the time to put your loop, your metal loop, into the incinerator, wait for it to get hot, then take it out, wait for it to cool. Instead, you just grab a brand new disposable loop. So again, it depends on the lab you're working in, whether or not you'll be using reusable metal inoculating loops or these single-use items. Well, the next item that we can look at is the disposable cotton swab. Those are very useful in the microbiology lab. They come in these sterile packages. There's usually 2 swabs in the package. You can only open these packages at one end. And again, just like with the loops, that allows you to open the package at the end opposite to the swab. So if you carefully manipulate the package here so that the opening is a little bit wider, you can pull the swab out carefully and you've got a nice sterile surface. So you can take culture material up with a cotton swab and use it the way you would use an inoculating loop. When you're done, you can simply toss that swab in the trash. Remember, transfer pipettes also come in these single-use sterile packages. When you open these up, you're going to first see the bulb end. And again, you want to open these carefully and pull them out carefully, but now the end that will touch your liquid broth is sterile. You can pull up a larger volume with these than you could with an inoculating loop, which makes them very nice. Very inexpensive, very widely available, very widely used, these transfer pipettes. Next on the list of tools is one of my favorites. This is called a hockey stick. Now you can buy a reusable glass hockey stick, but this is the disposable variety. Now watch what this stick is going to allow me to do. We've looked at how we streak cells across a Petri dish, an Auger dish. But sometimes we want to make a smooth coating of cells over the surface of that dish, and that's what the hockey stick does. See, you just swirl it around like the hands of a clock face, and you will spread your cells all over the surface of that plate, nice and evenly. One of my favorite tools in microbiology. At the end of the day, when we've finished our work, it's critical that we know where to dispose of our materials. And of course, when it comes to the materials that touch culture, we need an autoclave bag. This red bag has a biohazard symbol on it, and it clearly says on it, it's an autoclave bag. So this bag is where we put used culture-related materials, the things that have touched living bacterial culture or microbial culture. Our agar plates, our cotton swabs that we might have dipped into culture, our disposable transfer pipettes that may have drawn up culture, the cotton swabs, the hockey sticks, all of these things that have touched living cells that we cannot put in the incinerator because they're disposable. We're going to put in the bag. We're going to wrap that bag up with a tie, And that bag is going to go into the autoclave. Now, we haven't talked about the autoclave yet. We haven't talked about the process of autoclaving, but for now, understand that in the autoclave, a very high temperature is reached and a very high pressure is reached. And any cell and any spore that's inside that autoclave is going to be destroyed at that high temperature and pressure combination. Once the material comes out of the autoclave, it is ready to be thrown into the regular trash. All right, very good. So, anybody have any questions about anything that you saw in that video? You'll see I have gloves on. I just want to show you a couple of things that you saw in the video so you can see them in real time. I have an inoculating loop here. You can see the wire. The handle is made out of aluminum. These are very light. Here's that twisted wire with the loop on the end. Remember, these are very flexible, so I can play with it and get it how I like it. We also have disposable inoculating loops. See if I can do this. Disposable loops, again, single use. You don't have to go to the time of sterilizing this thing in between transfers, which people like, but they're single use. So you use it and then it gets discarded. So some people prefer not to use them. And I also have a couple of plates to show you. Here's an Auger dish that looks very similar to the one you saw in the video. The color of the auger in the bottom of this dish is a very light sort of beige color. So it almost looks like there's nothing in here. Yeah. But we melt and pour the auger into this dish, and we can add whatever we want into the auger in terms of nutrients to feed the bacteria while they grow. Remember, we always keep these upside down because we want the condensation to be able to drip off of the auger and then down onto the lid so it doesn't interfere with the cells. I want to show you, though, there's no bacteria on this dish. This is a sterile dish. But I want to show you how fragile agar is. I told you in the video that you don't want to ever try to work on a dish that is completely upside down because you're likely to poke through the agar. You always want to work at an angle. Or, as I showed you in the video, you also have the option to add the lid like this if it makes you feel like you're getting more protection from contamination. But it's perfectly fine just to take the lid off and keep your dish at an angle. But I want you to see how soft agar is. If I push too hard on this agar, I'm just going to ruin it. I'm just going to scrape it and ruin it. This is a second plate I have right here, which has a very dark agar in it. This is called eosin methylene blue agar. We're going to talk about it later in the semester, but if I'm not careful and I push into the agar, I'm going to break the agar. I'm going to tear it. It's not a stiff material. It's solid. It is solid, but it's not stiff. It's the consistency of Jello. It really is. It's the consistency of Jello or gelatin, if you'd rather. And if I push into it, I will just go right into it. So I always have to work just by sliding over the top of it with my loop. Plenty of us accidentally, we call it gouging the agar. Plenty of us gouge the agar when we're working. It's not the end of the world if you gouge the agar. It's just that whatever cells you might have pushed into the agar there may not grow very well for you. But again, the most common containers that we use in a microbiology lab to grow microbes purposefully are broth tubes and these plates, these agar plates. So it makes sense for us to learn how to handle them with aseptic technique so we don't accidentally contaminate them and thwart ourselves from getting our work done. Any questions? Any questions about what you saw in that video at all? Feel free to type into chat if you want, or you can unmute yourself if you want when you have a question, no problem. All right. Well, what we're going to do now is switch gears a little bit, and we're going to talk about a very, very popular differential staining technique called gram staining. If you remember, we said simple staining procedures use one stain, and they put one color onto the slide. And remember, we said if it's a direct simple staining procedure, it's going to stain the cells that one color. If it's a negative simple staining procedure, it's actually going to stain the background and leave the cells unstained. Now, the other type of staining techniques that we have are called differential techniques because they don't use one stain, they use multiple stains. And each one of those stains has a different color. And each one of those stains is going to stick to different materials in the cell. So we're going to end up with different colors on the slide depending on what's on that slide, what kind of microbe is on that slide, and so on. So the most popular differential staining technique that we use is called gram staining. And gram staining got its name because of the fact that the person who described it, who discovered it, if you will, and made it available to everyone else, had the last name of Gram. So we give that word gram a capital G, and we call it gram staining. So I'm going to pull up a new set of slides for us. If you give me just a sec, I'll try again. Nope, apparently it's not going to let me do this. Hold on just a sec. Sometimes I really don't like this. Okay. Hopefully you're seeing a slide that says gram staining on it. We're going to go ahead and get started now with these slides. So our objectives for this exercise are to first talk about why. Why do we use gram staining? What is it that we're trying to see on the slide with this gram staining procedure? We're going to talk about the role of each one of the different stains that we use in this procedure. We're going to talk about the actual procedure, how you do it. Then I'm going to show you how to do it. And then we'll actually take some time to look at some gram stained smears. And finally, we'll finish up talking about some of the limitations of gram staining. Again, very, very popular method in a microbiology lab. Gets done all the time, every day. But it does have some limitations that we need to be aware of. So first things first, why do we do gram staining? This is about bacteria, this particular differential staining technique. When we think about all of the different bacteria that we encounter in the world and in the microbiology lab, we can divide them into two very broad groups, one of which is called gram positive and one is called gram negative. Again, based on the results you get when you stain these cells with the gram stain procedure. This difference, the difference between what makes a bacteria gram positive versus what makes a bacterium gram negative is all about the cell wall. So some bacteria have a very thick cell wall, and other species have a very thin cell wall. And we can see the difference between a thick and a thin cell wall using gram staining. Remember, the material that's found in the cell wall of a bacterial cell is called peptidoglycan. It's a carbohydrate. We don't see it anywhere else in the natural world except in the cell walls of bacteria. It's very, very strong. We can stain it differentially. In other words, if there's a thick layer of peptidoglycan in the cell wall, we can stain it one color. And if there's a thin layer in the cell wall, we can stain it a different color using this gram staining procedure. And again, when we finish gram staining, we can determine whether or not this particular type of bacteria is gram positive or gram negative. Now, we often report our gram staining results when we're writing up a laboratory report. We report it as the gram status. So we will say, for example, that a particular bacteria that was found in, let's say, a urine culture has a gram status of gram positive coccus, for example, or perhaps gram negative rod. Notice that I said whether or not the organism was gram positive or negative, and I also gave you the shape of the cell. So those two things make up what we call the gram status of any bacterium. It's gram positive or gram negative result plus the shape of the cell. So when we get samples into, say, a diagnostic lab in a hospital setting, one of the very first things we do is we grow these bacteria in culture, as we've been talking, and we then determine their gram status because it's a feature of a bacterial species, what its gram status is. In other words, if I know that I'm dealing with, say, a gram positive coccus, I already know a lot about the organism. And I can plan what other tests I want to conduct to figure out exactly what species of bacteria is in that urine sample. So we learned about the cell wall in lecture. As you're looking at the prokaryote material this week, we also talk about cell wall structures. On this slide, you're looking at a drawing of the difference between the cell wall of a gram-positive bacterium and a gram-negative bacterium. Now you have to use your imagination a little bit here. At the bottom over here on the right, this represents a little piece of the membrane around the bacterial cell. So if we drew the whole bacterium, it would not fit on this slide. It would be much bigger than this, but we just cut a little sliver of the membrane and the cell wall so we could look at it in this picture. So you can see there are heads and there are tails and there are heads. This is that lipid bilayer of membrane that surrounds every living cell. This out here represents the cell wall. These purple circles represent molecules that make up peptidoglycan. And the artist has drawn 4 layers here to represent, a nice thick layer of peptidoglycan material surrounding this cell. So this is the outside area of the cell, and this would represent the inside area of the cell. This would be where the cytoplasm is, and the DNA and the ribosomes and so on. What's facing the outside of the world is this cell wall. And it's, as you know, one of the things it does is it protects this cell from the dangers that might be out in the world. Nice thick layer of peptidoglycan. Now I want you to compare it to what we would see if we took a little sliver off of a gram-negative organism. Here's that cell membrane again. Again, remember, we can call this the membrane. We can call it the cell membrane. We can call it the plasma membrane. This artist has labeled it as the cytoplasmic membrane. All of those things mean the exact same thing. the membrane that surrounds every single living cell on this planet. So this would be the inside again of the cell over here. This would be the outside world over here. So if we go from the membrane outwards, we again bump into some of this peptidoglycan. But this time, there's only a thin layer of it, a very thin layer of it. It's there, and it is protective, but it's not nearly as thick as what we see in gram-positive cells. Now, I want you to notice that there's something else out here around gram-negative cells, and that's the outer membrane. Gram-negative bacteria have a second membrane. They have their cell membrane, their plasma membrane, and then outside of the peptidoglycan, they have a second membrane, an outer membrane. Very strange. I also want you to notice these, what look like little green trees that are sticking off that outer membrane. These are molecules of that lipopolysaccharide material that we talk about in the lecture. We shorthand that long name to LPS, lipopolysaccharide, or LPS. Remember, this material that we find sticking out of the surface of gram-negative bacteria, this helps strengthen all of this structure that protects the cell. But we have to be knowledgeable about LPS as humans because we have a lot of gram-negative bacteria living inside our intestines. That's normal. If a lot of those gram-negative bacteria suddenly die because we have some kind of a disease or an injury to our intestine, if a lot of those cells suddenly die and the cell breaks apart, this LPS can be very toxic to us. It gets absorbed into our bloodstream and it's toxic. It's toxic to animals. It can make you very, very sick. And that's why some people who experience a sudden die-off of bacteria in their gut, again, due to disease or perhaps injury, that's why those people can go into shock very quickly because of this toxin called LPS. It's not toxic to the bacteria, but when it gets set loose, because these bacteria start to die, it's toxic to us. So we're going to take advantage of the fact that gram-negative bacteria have a thick layer of peptidoglycan. Gram-positive bacteria have a thick layer of peptidoglycan, and gram-negative ones have a very thin layer. We can stain those differently, differentially, in other words. So there are 4 separate chemicals that we're going to use to do this procedure. This is a direct staining method because we will be staining the cells, not the background. So we could call this a differential and direct staining procedure. Now, in addition to a couple of stains, there's also a couple of additional chemicals that are used, including one that's used to do some decolorizing and counter staining, and we'll explain those terms as we go. So here's the list of chemicals in the order that we apply them. The first one is called crystal violet. We actually saw an image in the simple staining lab of some bacteria that had been stained with crystal violet. Crystal violet can be used as a simple stain, but it is also used as part of gram staining. We consider crystal violet to be the primary stain because this stain is going to stick to the peptidoglycan. It's going to give it a bluish, purplish type of color. And again, we'll see some cells later on that have been stained through gram staining. The second material that gets applied in gram staining is not actually a stain. It's a special formulation of iodine called Graham's iodine. This is not regular iodine that you would find in a chemical lab. This is iodine that has been modified so that we can use it as what we call a mordant. Now, if you're not familiar with this term mordant, anytime you dye something, whether you're dyeing a bacterial cell, or perhaps you're dyeing your hair, or you're dyeing some cloth, anytime you apply a dye to something, you also apply something called a mordant. And all a mordant does is help that primary dye, in this case crystal violet, helps it penetrate and adhere to the material you're trying to stain. So Graham's iodine serves as a mordant in Gram staining. It's going to help the crystal violet penetrate the peptidoglycan and adhere to the peptidoglycan. Next on the list is our decolorizer. Like the name suggests, we're going to use this chemical to help remove some of this crystal violet, which might seem a little strange, but it'll make sense in a minute. The decolorizer that we use for Gram staining is just an alcohol. You may know that if you apply alcohol to things that have been dyed or stained, it can help pull some of that stain away. And finally, we have another dye, another stain called Safranin. You saw some slides in our simple staining lab that showed cells stained with safranin. Safranin turns cells a pinkish red color. Now, in Gram staining, safranin is the counter stain. And what I mean by that is we're going to stain everything purple-blue. Then we're going to remove some of that purple from some cells. And we're going to add another color, a counter stain. And that's the safranin. So it's stain, mordant, decolorize, counter stain in Gram staining. On this slide, you can see what's happening at a cellular level during Gram staining. So on the left side of the slide, we can see a drawing of a coccus-shaped bacterium. And this one happens to be Gram positive. Over here, we have a rod-shaped bacterium drawn, and it happens to be Gram negative. So they're telling us that a smear was made, 2 smears, this one and this one, and they were both heat fixed, and now we're ready for Gram staining. So remember, the first stain is crystal violet. Crystal violet gets applied to these cells for a certain amount of time, and we'll talk about that. And it stains the cells this purpley blue color. Then we're going to add our mordant, which is Graham's iodine. Now, the artist has just left them a purple color, but purposefully changed the purple a little bit. That's just to show us that this iodine is doing something. It's helping the crystal violet penetrate and adhere. So it's still purple, but the artist is showing us just a slightly different purple color. Now we're going to decolorize. We're going to add some of this alcohol solution onto our smear. And this is where the important thing is happening. You'll notice that on the Gram positive side, these cells that are Gram positive, they're going to stay purple after decolorization. Remember, we said Gram positive cells have a very thick layer of peptidoglycan around them. And it turns out that our decolorizing step is going to be limited in time. So we're only going to do it for a certain number of seconds. It's not going to be enough to take all of the crystal violet out of that nice thick layer of peptidoglycan. So those cells are going to stay purple. But look what happens to the Gram negative cells. When we decolorize Gram negative cells, because the peptidoglycan layer is so thin, we're going to get all the purple back out. Now we're ready for our last step, which is to apply the safranin, which is a counter stain. The gram-positive cells, they're not going to pick up any of this pink-red color because they're still stained with crystal violet. They're still stuck with crystal violet. The crystal violet is still adhering to the peptidoglycan. But those gram-negative cells, they're going to turn that pink-red color. So at the end of gram staining, any cell that is gram-positive, any cell that has a thick layer of peptidoglycan is going to be purple under the microscope. And any cell that has a thin layer of peptidoglycan around it is going to be pink-red under the microscope. Now we have a way to look and visually tell whether this bacterium that we're looking at is gram-positive or gram-negative. Any questions? Any questions about any of this terminology or why we're using any of these stains? What's happening at a cellular level? Now, this is where I want to tell you, gram staining has a little bit of an art to it. It's not a difficult technique per se, but you can mess this up if you're the person doing the gram staining. It turns out that if you don't do the decolorizing step correctly, and again, I'm going to demonstrate for you how to do it. If you don't do it correctly, particularly if you don't do it, or if you do it too long, you leave the decolorizer on for too long, what's going to happen is you're going to remove all the purple from all of the cells. So it is possible to mess up a gram staining procedure, to accidentally decolorize for too long, and you're going to end up with all the cells being pink, even though some of them are gram-positive. So I just want to mention that because, like I said, there's a little bit of an art to gram staining. You have to know exactly how long to leave these stains and other chemicals in contact with the cells. All right. I'm going to jump back in here. Now, this is a very busy slide. Don't try to write all this information down. Remember, you have these slides available to you if you need to come back and pull this up. This is simply the procedure for gram staining written out completely. I'm going to talk us through this, though, so you don't have to write it all down right at this second. Remember, the first thing you have to do is have a smear made, and you have to heat fix that smear. Always, always, always. If you want to stain bacterial cells, you have to make a smear, and you have to heat fix it. So we're going to apply the crystal violet, as we said, and we're going to let it sit. And specifically, we're going to let it sit for 30 seconds. Then we have to get the crystal violet off the slide. So we're going to gently, gently rinse it off, and we're going to use distilled water. That's an important point. Distilled water, like the name suggests, has been distilled. It's had all molecules, including cells, removed from it. So it's pure water. It doesn't have any microbial cells in it. That's important because we wouldn't want to accidentally put a microbe onto our slide that doesn't belong there. Remember, tap water can have a few cells in it. It's just the way it is. Now, once the crystal violet is washed off, we're going to put the grams iodine on. We're going to let that sit for 30 seconds as well. We're going to rinse it off with distilled water. Then we apply the decolorizer. We're going to decolorize for 10 seconds, only 10 seconds. And we're going to apply the decolorizer drop by drop. Again, I'll show you this in just a moment. Rinse with distilled water. Then we apply our safranin and let it sit. Now, safranin needs a little bit longer than the other chemicals do. Safranin needs a full minute. So we'll let the safranin sit on the slide for a minute. Then we'll rinse it off with distilled water and we'll blot the slide dry. All right. Let's walk through gram staining now with some pictures. On this slide, you can see the face of an incinerator. You can see I'm going to get my loop into the incinerator to sterilize it because, of course, I'm going to be moving cells around. I need a sterile loop to do that. In this picture, which you've seen before, I have my culture tube. I've put my sterile loop in to gather some cells. I have a gloved hand, of course, because I'm handling a culture tube. I'm keeping the lid in my fingers. I'm not putting it down on my desktop because I always assume that my desktop is contaminated. I have the tube at an angle because I want to prevent microbes from falling into the tube while it's open. I'm going to make sure that I mix up my culture before I draw out of it. And I'm going to draw from that middle area in the culture tube. Remember, I'm going to label my slides. Remember, always, always label your initials, the date, the name of the organism. Here I have my inoculating loops. I'm going to use to put those cells in the middle third of this slide. And you can just see the motion, the circular motion again that we use when we apply cells to a slide to make a smear. Your circles just get bigger and bigger and bigger as you go. You start in the center, and then you make these ever widening circles to spread out your cells. Remember, we want a thin layer of cells on our slide. Now, I put this video in. Remember, we said it's super important that we dry smears before we heat fix them because we don't want to accidentally aerosolize or send cells up into the air by heat fixing a wet slide. Remember, if you were to aerosolize microbial cells, people in the lab could inhale those cells, which would not be a good thing. So you can just leave your smear on the desktop to dry. We said you can also put your smear on a slide warmer, remember the hot plate sort of device, to dry. The other thing you can do is just gently do this. Remember, microbes can't jump, okay? So anything that I have on this slide is not capable of jumping off. This simple, gentle waving back and forth is not going to be a problem in terms of microbes getting into the air. Now, notice I'm holding the slide at an angle because I do want to try to keep things from falling down into my smear. But a little gentle waving in the air is not going to cause a problem, and it might help your slide dry quicker. Now I'm ready to heat fix my smear. Remember, you always want to use something to hold your slide. Don't put your fingers up next to a hot incinerator or an open flame. Oftentimes we use these wooden clothespins because they're very inexpensive. So I'm using the clothespin to hold the slide so my fingers stay away from this hot metal. I'm going to lay the smear on top of the metal and hold it there for 20 to 30 seconds. Now I'm ready to stain. Now you'll notice I have 3 slides laid out here, and I wanted to do that because when you're working in a microbiology lab, it's very rare that you're ever making a single slide. You know, if you come into work and you work in a hospital lab, for example, and your job is to examine patient samples for evidence of microbes, you're rarely going to have a single sample. You're going to have several samples. So it's very common for us to have to make several slides all at the same time. And it's perfectly possible for us to make those all together. So the first thing we do is we make each individual smear. You'll notice I drew a circle with Sharpie on these slides. Again, some people like to do this so they know exactly where their smear is. Um, then I heat fixed all 3 of these slides, and now I'll stain all 3 of them at the same time. So I'll have everything done and ready to examine. What you're looking at on this slide is a very low tech, very inexpensive piece of equipment that we call a staining tray. These come in all shapes and sizes, but they're usually homemade. Very, very easy to make, very inexpensive to make. You can see I've just got an inexpensive plastic food container here. And I've taken 2 zip ties, 2 stiff plastic zip ties. I've poked holes through the plastic box and I've run the zip ties through. Now, what this does is it creates a rack, essentially, where I can lay down my glass slides and they will be suspended over this box. Now, the reason we use staining trays like this is because, remember, stains are hazardous. So we have to collect all of the stain that we use so that we can then put it into a hazardous waste bottle. So by working over this little plastic tray, I can gather all of the different stains I'm using, all the rinse water, all of the decolorizer. I can gather all of it into this container and I can then transfer it into the hazardous waste container. So I've laid 3 slides down over my slide tray and I'm ready to go, my staining tray, and I'm ready to go. Now, gram staining is such a popular procedure that there are kits that you can buy from commercial supply houses, and that's what you're looking at here. I can go online to any one of the big commercial suppliers. This one happens to be Becton Dickinson. And I can buy a gram staining kit. They're generally supplied in these large plastic bottles. So you can see this is the crystal violet. Here's the grams iodine. Here's the decolorizer. And here's the safranin. All 4 of those will arrive together in my kit. It's about 1 liter, I think, of fluid in each one of these bottles. Now, if you're a busy microbiology lab, if you're working in a very busy lab, you can go through these bottles relatively quickly. If not, they last a long time because there's a lot of volume of each one of these chemicals in here. On this slide, you're looking at step 1 for the gram stain procedure. I've got my 3 slides right here sitting again on the zip tie shelf over the staining tray. And you'll notice what I'm doing is I'm placing crystal violet over each one of the smears. Notice that I am just flooding that smear with crystal violet. You really have to make sure that you've covered the entire area of the smear with crystal violet. You have to make a puddle on your slide. Now, these stains, these gram stains, these are not expensive. So it's not a big deal to put a little bit more stain on here than you need. You can see over here on this first slide, I've really flooded this one. Okay, so make sure you have the entire smear covered in these stains. Once I have the crystal violet applied on top of the smears, I'm going to let it sit for 30 seconds. Now, in gram staining, that time, that 30-second time period is not strict. Now, what do I mean by that? In some protocols that we do in science, the time intervals that we have to use are very, very strict. In other words, when they say 30 seconds, they mean 30 seconds. But in gram staining, it's very liberal in the sense that, if you left it on for 35 seconds, it's no big deal. If you left it on for 28 seconds, it's no big deal. Approximately 30 seconds is just fine. Now you'll notice I've picked up the slide again with my clothespin. And now I'm going to rinse the crystal violet off into my staining tray. I'm using distilled water. I'm just going to squirt the water over the slide using a transfer pipette to get that crystal violet off. Now I want you to notice something here. Look into this Sharpie area here, the Sharpie circle. This is where my smear is, and I just stained it with crystal violet, but my slide doesn't show any purple on it. Remember, if you make a smear correctly, you're not going to see stain until you look under the microscope. Remember what we're doing here. We're staining the cells. We're not staining the slide. We're staining the cells on the slide. And since we made a nice thin layer of cells, we're not going to see them as looking purple. They're microscopic. So it's perfectly normal not to see anything while you're getting your slides ready during gram staining. In fact, if you finish rinsing at this point and you see a big purple area on here, it means your smear is too thick. You haven't spread out your cells enough. Now we're ready for the grams iodine. Remember, this is not a stain. This is called a mordant. And mordants help stains penetrate and adhere to the material we're trying to stain. I'm going to make a nice big puddle of grams iodine on all of my slides here. And I'm going to let that sit again for approximately 30 seconds. Now I need to rinse off the grams iodine. Again, into my staining tray, so I collect all of it so it doesn't go down the drain. Now it's time for decolorizer. And this is the step that gives students a lot of frustration because, again, this is the step that you can do incorrectly. I'm going to draw up some decolorizer in a transfer pipette. I'm going to drip it over the smear, drop by drop, for 10 seconds. Okay. You'll notice that as I drip it out here, I kind of move the pipette back and forth so that I'm sure to drip over the whole smear. But I'm just putting droplets of decolorizer on here. I'm not doing what I did with the stains. I'm not putting a big puddle of decolorizer on top of this. All right, just drip, 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 10 seconds. And that's all I need to do. Now, I've just put alcohol on my smear, and I've also said that decolorizer can be left on for too long. So it's very important that you rinse the decolorizer off, just like you did with the other chemical stains. So you have to rinse off your decolorizer, just like you did with your crystal violet and your grams iodine. Now I'm ready for the last step, which is the counter stain called Safranin. Make a nice big puddle over the smear. And remember, unlike the other ones, Safranin gets left on for about 60 seconds. So about twice as long as the other chemicals. Rinse for the last time. And now I'm ready to blot. This is that bulbous paper that it's just a very thick, very absorbent paper. I lay the slides down. I fold the paper over and I just gently blot the slides to get them as dry as possible. The last thing you want to do is rub a slide because you might rub your cells off. And here are my 3 stained slides, gram stained. Notice that there's some smudgy purple on these. That's normal. Notice too that some of my Sharpie circle has gone away. Decolorizer can remove Sharpie. So if it's important that you keep your circle because you're learning and you're not sure where to find your smear, you can use a different marker. You can use something called a grease pencil instead of a Sharpie. The alcohol won't remove grease pencil. Now I'm ready to examine my results. So, questions about this procedure. I always want to work over a staining tray. Anytime I'm doing any staining, including gram staining. And the reason is I want to be able to keep hold of all of that waste material so I can dispose of it correctly. It cannot go down the drain. All right, good. All right, on the screen, you should see a slide that is being looked at under the microscope. This is a slide that has two different kinds of bacteria on it. So this is not a pure culture. This is not something we grew up in the lab, but rather something that came perhaps from a patient or perhaps from the environment. We're using the 100x objective lens here. So our total magnification here is 1,000 times. Remember, I can see bacteria under the 40x lens, but I can see it best under the 100x lens. Now, I can clearly see two different colors here. Yeah, I can see a purple color and I can see a pink color. I have two kinds of bacteria here. One of them is gram positive and one is gram negative. The gram positive organism is the one that's stained purple. The gram negative organism is the one that's stained pink. So if I was looking at this slide after I gram stained it, and I had to write up a report about what was on it, and I had to report the gram status, I would say I have a gram positive coccus. You can see these cells are spherical or circular. And I have a gram negative rod. See that elongated cell? That pink red cell is rod shaped. Yeah, everybody see that? Now, clinically, again, in medicine, a lot of gram positive organisms happen to be coccuses. They happen to be cocci, as we say. So a lot of the gram positive things that we run into as pathogens tend to be cocci. They tend to be these circular or spherical cells. Think about Staphylococcus or Streptococcus. A lot of them, gram positives, happen to be cocci. And similarly, a lot of the gram negative pathogens that we run into in human medicine happen to be gram negative rods. There are things like Salmonella and E. coli. There's one called Pseudomonas. Those are all gram negative organisms, and they all happen to be rod shaped. So this is not an uncommon result, but it's very important to remember that there are definitely gram positive rods, and there are definitely gram negative cocci out there. It's just that when it comes to pathogens that we run into as human beings, a lot of times the cocci are gram positive and the rods are gram negative. Not always, but a lot of the time. All right, take a look at this slide. This is not a pure culture. There are two different organisms growing. So maybe this is a patient sample. I want you to tell me what you see. You can type it into chat, or you can unmute yourself and give it a try. First of all, gram positive, gram negative. Do you see rods? Do you see cocci? What do you see? I got a vote for gram-positive and gram-negative rods. Very good. That's good, Brooke. Yeah, we've got two kinds of rods on this slide, right? We've got some rods that are staining pink, and we've got some rods that are staining blue or purple. So I've got two organisms here. One of them is a gram-positive organism. It's rod-shaped. It must have a very thick, as a gram-positive organism, that tells me it has a very thick layer of peptidoglycan in its cell wall. It's also rod-shaped. It's a gram-positive rod. I don't know yet what it is. I don't know the species yet because I'm going to need to do more testing. But I've already cut down on the possibilities just by identifying it as gram-positive. I have a piece of information about it that I can use to help identify it. I know its gram status. There's another organism on here, and it's a gram-negative rod. Again, I don't know exactly what species it is yet. I can't know that until I do some more testing. But I know its gram status. And so I've already learned something about this organism. I have a piece of data about it that I can apply in my investigation to help me identify what it is. Any questions? Anybody not see the two rods on that slide? I didn't mention it, but that slide was also under the oil immersion lens. When we're examining our gram stained slides, we're going to want to go all the way up to the 100x lens, the oil immersion lens, because that's going to give us the best view. All right, very good. When we get together next time, what we'll do is we'll look through a whole lot of examples of gram stained slides so we can get some practice. We'll also look at some clinical specimens that have been gram stained, and we'll see if we can guess what it is, what the gram status is for the organism in the samples. Okay, we'll do that when we get together on Friday. Does anybody have any questions? All right, before you go, if you haven't already, be sure to type your name into the chat. If you have a nickname that you use, can you type that in for me as well? That's helpful for me. All right. Thank you. So we're just about out of time. I'm going to let you go. Remember, your lecture 1 exam grades will be back by Friday, early afternoon, perhaps before then, if I get time. But certainly by early Friday afternoon, you'll have your grade back. So keep a watch out for those. Don't forget you have the prokaryotes material this week to do, the lecture quiz, and of course your lab homework. And we'll see you on Friday. Okay, enjoy the rest of the day.
Thank you.
See
Thank
you
you.
next time.
Thank you.
You're welcome.
Excuse me, Professor?
Yes, yes.
Did you get my assignments?
Yes, I did.
Okay.
Is this Rue?
It is, Ro, yeah.
Yes, I certainly did. So whatever you did, it worked.
Yeah, so I usually do them with Yashi, so I'm a little confused why they're not going through.
I wish I could help you more than I can. It's all that technical stuff regarding Blackboard. I can't help with because I don't have access to it. But if
I
you,
don't think it was Blackboard. Like, I think it was also like I was doing something wrong my end. Like maybe I wasn't submitting it or saving it.
yeah, that could be. That could be. You know, that's one of the drawbacks with Blackboard. You have to take a couple of steps that, you don't really think you would have to take, but you
Yeah.
have to make sure you click that button after
Okay.
you've done your work to submit.
Yeah.
And it's easy to forget that. So,
Yeah.
but I'm glad you got it figured out because
Yeah.
everything came in this
Thank
morning.
you so much for telling me because I wouldn't have known.
Oh, yeah, absolutely.
Yeah.
Absolutely.
It's usually like, well, this class is a little bit like on the easier side for me, so I don't really pay that much attention to it than like I do with organic chemistry.
Oh, yeah, sure.
So
Yeah,
once the
absolutely.
assignment, I think like, okay, I'm done with this class, so then I move on to the next class, and I don't think I would have noticed until you told me.
That's
that
why I do that. I try to always do a survey, who's handed it in, who hasn't, and if I see that something's missing,
Yeah,
especially if I see more than one thing missing, I try
yeah,
to reach out because I know that, like you just said, a lot of times we do the work, we hand it in, and then we don't think about it.
yeah.
Yeah.
All right. Thank you so much.
Oh, you're very welcome.
Thank you.
We'll see
Bye.
you next time.
See you next
Bye-bye.